Thank you very much, Beppe, for agreeing to give a Build a Cell seminar, and you can get started.
All right, thank you very much, Kate, for having me here. It looks like you had some really cool speaker. Hopefully, I will maintain the series up. And what I'm going to tell you today is a story of how we try to copy and mimic biological emergent properties, but more important, how we can translate whatever we learn into application, etc. Um, just to start, I always prefer to start and get this out of the way because it's always nice to acknowledge that people really do the work and they really make sure that we have enough data and nice information to pass on. So my research group is based in Barcelona. I'm the group lead of the Molecular Bionics Group, which is part of the Institute for Bioengineering Catalonia. I have a very nice and interdisciplinary research group comprising mathematicians, physicists, chemists working together. I have the pleasure to co-lead this group with Professor Lorena Ruiz Perez, who is also a professor at the University of Barcelona in the Faculty of Physics. And I also have a very fruitful collaboration with Professor Xiao Tian from the West China School of Medicine, where I'm also affiliated. And we have this very active collaboration program where many students from his lab and my labs come and go and move from Chengdu to Barcelona. A few years ago, I moved from London, where I was based at UCL, and I still have a couple of students, Joe and Melis, about to finish. I think in a couple of months, I will read out, defend the thesis. And as a sort of disclaimer, also founder of a company by an artist, which is in Cambridge. So also trying to develop gene therapy. Anyway, this is kind of the people involved in the work. Let me try to, again, with a sort of a top-down approach. My research group is a mixture of physics, chemistry, and biology. Our main objective is to design advanced therapies. We do this either by creating the delivery system, but most importantly, recently we are actually discovering a way to create completely new drugs. And the way we do that is, of course, like everybody else, we start from the disease side, from the clinical point of view, understand how disease evolve, what are the targets, etc. But then we quickly deep in understanding how things move inside a disease area in the body and understand how biological transport can be either bypassed or exploited to really get stuff into the right place. And for doing that, we exploit and try to take as much information from complex physics and non-equilibrium statistical physics, particularly the phenomenon, emergent phenomena, which is for us is important. I will try to explain this more and more in detail later on. And as we gather all this information, we feed all of this into chemistry and in particular into what we call molecular engineering. We try to design those devices in order to get that advanced therapy effect that we after. Um, to start, I want to go right to the beginning and very philosophically here, try to put some sort of context in, and at least my opinion, how life is constructed and what we really need to take into consideration. We try to copy and mimic it. It's not difficult to do this graph, which I show you, simply by looking at size and number of components. You quickly go from molecules all the way to up to organisms, passing through, of course, proteins and DNA or polymers, cells, tissue, etc. But what is very important to acknowledge is that this number arrangement is actually much more than we think. It's from a chemical point of view, it's very easy to assume that every water molecule that comprise our body are identical, to some extent anyway, without going into quantum mechanics. And then definitely for the same species, at least nucleic acid, RNA and DNA, are identical. In other words, the RNA that encode for a protein is the same as the other, etc. But already as we move from species to species, there are many differences in sequencing, and even though you more or less get the same structure. And of course, as we go into the translation exercise and the RNA is converted into proteins, yes, again, from the same species, we can assume the two proteins are identical, but these proteins are very short-lived as a single amino acid component, and very often they get what is called post-translation modification. And this post-translation modification already creates an interesting diversity exercise, and many proteins actually diversify very quickly. So the same protein, although it seems identical, they're not entirely identical, not at least molecule by molecule. And as we go up into the complexity and we go into cells, even clone cells, cells that are genetically identical, they don't have the same internal state. The reason I'm trying to point forward this diversity in chemical equivalence in biological system means there is a huge amount of arrangement and information flow that biology includes in order to create a complexity that then is a living system and then becomes alive. Um, then another important element is how these elements interact with each other and how this interaction becomes much more complex in terms of linearity. Not often, almost never you get an output that change linearly with the input. You always have these non-linear processes where you have very narrow changes into the input and quickly your output change massively and catastrophically, almost as a phase diagrams, as a phase transition type of changes. But you also often get non-linear behavior where you have non-monotonicity with one peak or even multiple peaks, etc. And this is very important and very often observed in biology, which means you need to really be careful in identify these sort of regimes and where everything happens, especially when you try to make drugs altogether, and this is again my view, if I put all of these interaction, number, arrangement, and size, you start to see the emergencies on life. And effectively, as you go in more complex and complex number arrangement and create more and more inflammation, you start to create complexity behavior, and not just the alive cells, but also coordination of tissues and emergent organs. And most importantly, probably the most beautiful example is our brain, where conscience emerge as a consequence of that. The price for this, however, is that now you have a time factor to take in consideration everything as an aging element. So in other words, even the more simple, the simplest protein tend to have some sort of expiry date, and after a while, time will be just transforming that into something else. And this is very important when we deal with this complexity. So what I'm trying to do is really trying to explain as we go into complexity and living system, you need to really embrace the idea that as you put things together, you start to have a very surprising interaction emerging. And this is called emergence in living system. I'm going to give you two examples here. And I'm going to give you particularly how we can use this example to then understand biology as well as to design new therapies and new biomaterials. I'm going to start with multivalencies and how we can use these for designing drugs. In all of them, everything is embedded into using a very specific way of making materials that somehow copies the complexity of life. And people do things using liposomes and nanomaterials from inorganic sources, etc. Our lab likes to work with block of polymers, things that are same as most biological materials. Once in water, they are able to self-assemble into very, very specific structure. We work with things that have an hydrophobic hydrophile in nature. They are polymeric in nature. And then as you change the ratio between those two components, you can form spherical micelles, cylindrical micelle, or disc-like micelle, or even vesicle that we call polymerisms. So we've been working a lot with this type of structure from a material point of view. I'm not going to spend any information about here. I'm not going to tell you how we synthesize, etc. If you want to know, please be in touch and I can point you out the relevant literature. Also, more important how we characterize them. This is also a very critical element. The other things I'm going to say quickly on this type of materials is that you can complicate your complexity and introduce other elements by simply having, for example, a third hydrophilic component into the equation. You can start to see that this system will emerge as a combination of the interaction between hydrophilic, hydrophobic, and then the two hydrophilic elements. In this particular case, which is recently published, we show that if I make a tri-block with the ABC element, I end up with a complete Janus-like micelle where one side of the micelle is different from the other, etc. Or if I mix this di-block, tri-block element into a vesicle arrangement like polymerism, I can start to have some very complex patterns emerging as the consequence of the interaction between di-block, tri-block, etc. So this is again a very important element has to show how by simply modulating the number of elements at place and controlling the interaction between them, you can start to create some very interesting emerging property. Another important element to consider, and this is the reason why we work with the polymers, is that these systems are intrinsically soft. They are very, very almost like water-like in some sort of element and sort of mechanical properties. What I'm showing you is a simulation of a block of polymer micelle made by the blue hydrophilic polymer called PEG and an orange hydrophobic polymer called PLA. And again, this is important for us to understand how this softness emerge and what it means in terms of arrangement and structural properties, because of course, we need to know where everything goes, etc. More recently, and in collaboration with Lorena Ruiz Perez from the University of Barcelona, the co-lead of the molecular bionics group, we actually were able to even capture the softness in real time using a liquid phase transmission electron microscope. This is a normal electron microscope. Well, it's not really normal, but it's a sophisticated, it's a very powerful electron microscope where we encapsulate the sample into electrotransparent window like silicon nitride or graphene, and then you allow them, you can image them in liquid water without the need either to cryogenically preserve them or to remove the water by dry state. And what we see there is a single micelle, like I showed you before, but this time is the real capture of the images over seconds. And again, you start to appreciate the software and even capture the molecular properties. So this is a bit of a diversion just to show you how important it is to characterize your sample, because the very first task I need is now to have a very high level of control on my supramolecular arrangement. I need my system to have a very specific size, a specific shape, and most importantly, specific valencies. In other words, I want to build up my molecules from polymerizing certain molecules which are compatible. So I'm not going to go there too much detail, but one thing we use is metabolites from the body and co-polymerize them in order to form our components. The reason we use those is because we want them to degrade into something our body can metabolize, or in some cases to create even biological action by using certain metabolites that have a certain property. For now, what you need to know is these things, they self-assembly and control fashion into these sort of soft yet well-defined structures. And by simply mixing polymer which are completely pristine with polymer which have a certain ligands on the surface, you have a very high level of control on the final stoichiometry and induce the valencies of our system. Why this is important? Because we want to achieve multivalency. What is multivalencies? This is a very important element. And typically, without going into much details, when you design a drug, you always design with a target in mind. And this is something that now drug discovery and medicinal chemistry and biotechnology are doing extremely well. As soon as you have an information about your protein target, you almost, in matter of weeks nowadays, you can design a molecules that bind almost exclusively to that molecular target. And for those of you that ever done biochemistry, you probably design, you know them as a key lock model. It's a very common and a certain model. And typically this is represented as a reversible reaction where an on and off rate combine into an association constant, etc. And you can measure this interaction, and this is normally defining the drug property, one of the most important, called affinity. So how strong the drug binds and exclusively to its target. Just to show a very simple mathematical treatment of this, you can represent this process as a sort of a surface, a saturation effect where your cells with a receptor represent a given surface with a certain number of receptors, so binding possibility, and then the drug will simply saturate this, reaching a fraction of 1 that is represented as this theta 1. Now, the problem with this approach is if now I start to look at the diversity of cells. Let's say I want to target the cancer cells, and I know that receptor X is expressed in cancer cell, is associated with cancer metastasis or something to do with the disease state, and I want to make sure that my drug only reach the cancer cell and no other cells. This comes problematic in a system like a human body because the receptor that expressed in a cancer cell is never really unique to cancer cell. What you have there is a cells that express more receptor than other cells. Now, if I design my drug in a way that interacts strongly with the receptors, now whether the cell has one receptor or 100 receptors, the result is always the same. You always get full saturation on your surface. In other words, you always get interaction between your drug and your receptor. Of course, understandably, if your final objective is to kill the cancer cell, you end up killing cells which are no cancer, and therefore you have a lot of side effect. Nothing surprising here. Most of you heard how terrible is chemotherapy in cancer treatment, etc. This is a very common. Normally, what you do, you try to find a way that you compromise the damage with the cure and try to limit to the maximum the side effect. However, you always end up with losing materials and, of course, interacting with cells which are not your target. Now, one approach to do so, of course, if I have a very bad drug, it will never saturate it. I'm going to keep it there for references. One approach to change things is to create a multivalent type of interaction. In other words, instead of having a single ligand, I will join together more than one copy of the same ligand. Now, by doing so, I create a completely different type of interaction because now the single molecules that have let's say has a three ligands for the sake of argument, can interact with the receptor in many different possibilities. In other words, the possibility of association becomes higher than a single possible ligand. And so that creates an extra energy in place, which is totally entropic and is independent exclusively by the number of possibility or number of association state, this omega here. So to give you an idea, if I have a trivalent ligand with the one receptor, I will have the possibility of forming three possible arrangements, either the bond here, the bond there, or bond there. As I go into a system with three receptors, this combination increases considerably, and then the number can go simply as a 3 times 3, because you have one single bond possible at any given time. When you have a very flexible topology, this number increases factorially. And I'm going to go in not so much in the detail of the matter, but just to give you an idea, this number becomes 14, etc. Which means I can create energy by simply play with the numbers. So what I can do now, if I make an high affinity monovalent into a multivalent, nothing's going to change. It will make the interaction stronger, but I will always saturate my cell whether I have one receptor and a receptor. But now if I take that kind of low affinity, which I put there just as a reference, and I make it multivalent, this is something interesting because now I make my interaction completely dominated by the entropic contribution, so that the interaction becomes possible only when a certain number of receptors is reached. So before that, I have zero interaction. And it's exactly what we want, because now I make my system super selective to the number of receptors. Now, this is simply to explain this concept, as also known as super selectivity. And actually, it's very symmetrical. If I invert the graph, and instead of receptor number, I put ligand number, the same rules apply. Now, in this case, however, experimentally is very convenient, because what I can do, I can have my cells here, which I want to target, and the cell I don't want to target. And so creating different valencies into my nanoparticles or supramolecular object or whatever you decide to use, and identify the number of ligands where you only interact with the cell with the highest number of receptors. And this is great because it allows you to really have a very high level of interaction. Now, this concept was actually published in 2011 by, I forgot to put a reference, apologies for that, by a colleague in Cambridge, and it worked as long as your single bond is extremely weak. And that, of course, is very difficult, because if I show you again, sorry, if I go back to my slide here and I repeat the animation, the same animation, my low affinity, in order to be very, very weak, it has to be absolutely useless. And so this is physically impossible to reach by itself. So what we have done, we created a completely different approach in order to make the bonds weaker, using our polymer design. What we do when we attach our ligands to our block of polymer to form our supramolecular structure, we actually deliberately make the one that has the ligands with a tether much shorter than the pristine one, so that as this structure come together, and then approaches its target receptor, the interaction is not just simply the ligand's receptor, but also you have steric hindrance or interferences from the brush around. This allows us to make the single bond interaction weak enough to create a super selective regime, and therefore allowing us to make the interaction exclusively dependent on the number of receptors. So this was published now a few years ago. We also show that you can create synergistic combination by combining different type of receptor binding. And now we regularly apply this to many problems, in particular targeting the brain endothelial cell, which is part of the BBB. I will show you that later. And again, you create libraries of particles, you put your cells, for example, in this case, using macrophages and brain endothelial cell. I want to target brain endothelial cell. I need to identify the receptors they have. The ligands, they have exactly that kind of interaction only with the brain endothelium, and avoid those numbers which interact with everything. And this is good, but still not enough, because actually, if I take my multivalent drug and put it into the real world, and here what I did is did a simple artistic exercise where I creating a simulation of the blood plasma, putting together all the possible protein accessible there, and then what a cell membrane looks like in terms of typical composition of receptors, and not just that, by a lot of sugar like glycocalyx, etc. What you see there is a very crowded environment, which means our system not only has to interact with this receptor, it has to avoid the rest. And that is not a trivial task. So it's very important we create, first of all, a system that is very repellent to everything that we don't want to target. And then once we do that, again, we can play our selectivity games, but also understanding that you're going to have interactions which are repulsive, not only from your next to your target, but also from the environment around your target, like, for example, the glycocalyx, etc. Now, if we account for the sterics, it means the energy total now is binding plus this repulsion. So you have a two completely counteracting component of energy that basically means if we plot this energy again, I show you already that attraction increases or decreases with number of ligand receptors. But now if we account for the sterics in a simple and the most simplistic approximation increases linearly, now the total energy becomes non-monotonic, which means now if I go back to theta, I will have a binding which is controlled by, like I showed you, by superselectivity. But if I keep increasing the number of ligand receptor, then actually I get to a regime where it switch off completely. And this is because I will have too many elements at play. Now, these too many elements actually is not that many. In some cases, even with few ligands, you start to observe this non-monotonic behavior. Now, more recently, we've been playing with different way of steric control our ligands and observe exactly the either a behavior which is a typical on and off like the blue line here, which I showed you before already, or in this case when we get more sterics, you actually have this sort of beautiful non-monotonic behavior where we have this sort of range selectivity where things happen. So long story short, by simply putting together bonds, you start to create a complete new way of targeting cells. And of course, this multivalency topology can be arranged and constructed in a different way. You can create a single polymer. You can create a polymer stabilized nanoparticle. You can create multivalence with ligands, like I show you, multivalence plus brush. But remember also your receptors. And so information about your receptor are critical. And more recently, we discovered that actually some receptors do not have one binding site, they have multiple binding sites, and that complicates things and make even non-linearity in a more extreme way. So with range, which are start to emerge in a very, very narrow way. Long story short, the way that we design our things is no longer based on the single receptor, but rather on the composition of the receptors that the cell express. And this is in biological terms known as a phenotype, and we call it, as a matter of fact, a phenotypic targeting. And so this is great in order to get very high level of selectivity. Now, what else we can move multivalencies into place? It is a very simple implication. Again, just simply by creating a combination of bonds, a few things emerge. First of all, now the binding is always favored because the unbinding, as one bond is formed, the other one is, as one bond is broken, the other one is formed. So that effectively the K of the total system will be very, 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 very close to zero, which means these particles tend to spend a long time bound to its target. You also can exploit the multivalencies to cluster receptor together, and you can do so many interesting things. And more interestingly, you can actually cluster receptor in a way that you can manipulate the cell membrane in order to trigger a process which is very important in cells, which is called endocytosis. Again, this is a collective effect emerging from this combination of bonds. As you manipulate these, you can do so many things, including exploiting the catabolic machinery of the cells and creating, other opportunity. Again, I'm not going to go to the detail, but if you want to know more about this, please be in touch. Now, this process of membrane modulation and deformation is a beautiful physical problem. Actually, I saw that Lipovsky was one of your speakers. I'm pretty sure he presented you these models in much more details than I did. But basically, long story short, as a particle binds to a cell membranes, the liquid nature of these membranes means when you have a certain curvature, you actually can favor this deformation very, very well. And this basically means that you can create a control of this process by simply changing the size, of course, the binding interaction, and more important, again, the receptor concentration, so that you can actually make selectivity based on how these particles are processed. And more interesting, you can start to see even collective effect at the membrane level. This is a simulation that we done in collaboration with Angela Sarik from which was in UCL when we did this work. Now she moved to Austria in IST. And what I show here is basically particle approaching a cell surface. And then the particle is not strong enough to create a deformation by itself, but then as they bind to the cell membrane, and more particles arrive, and now a collective effect emerges. And now the deformation is no longer single particle driven, it's actually driven by multiple particles that line up together and create this beautiful tubulation event. And so you can map it out, etc. Now, why this is important? It turns out that in many cellular behavior, this tubulation process is a very critical element. And we discover actually, I'm not going to go into the biology of it. Again, there are a few papers and more. I forgot about to add that the newest work here is that cells that create the vessel that carries our blood in our body, and in particular the vasculature that feeds our brain, has a very clever way to use tubulation to recognize the cargo that needs to be transported across from the blood into the brain, and the cargo that has to stay inside the cell for the sustenance of the cells. And so we discovered that as a function of the valencies, you can trigger this sort of multivalent and collective endocytic effect and gets across the endothelial cells, and in this particular case, get into the brain. Or you can trigger a traditional conventional endocytosis where you end up into the catabolic part of the cell and get degraded or not. Turns out this is not just something that you can do with synthetic particles. You can also serve this into natural multivalent structure. And one of the most interesting one, amyloid beta. These are misfolded proteins that emerge as a consequence of a disease associated with the Alzheimer and dementia. These are proteins which are very insoluble. They start to aggregate into oligomer and fibers into plaques, etc. So inevitably, you have different valency as you go from monomer to fibers. And again, they also exploit this element. They can actually either go through this process called transcytosis through tubulation, or can get stuck into endothelial cells by endocytosis. And what is really interesting, actually, as this process is triggered, you not only change the biology of this membrane, because of course the receptor now is go from one side to the other, while in this case it get degraded. You also change how these receptors then manage endogenous protein like amyloid beta. So long story short, if I trigger, I use a multivalent drug like I show you that target these pathways, effectively I can create an activity where the endothelial cells become activated to remove more toxic species. And then long story short, I'm going to give you a quick example of that. We published this last year at the very end of the year. We show that if you have the right balances and treat an animal that is diseased to express amyloid beta and therefore then present Alzheimer and dementia, with the right balances, you trigger this pathway of recovery and therefore remove amyloid beta from on the brain in a single injection. 2 hours later, if we measure the A beta concentration into the brain, we see a reduction of about close to 40%. And the most importantly, the same amount of protein is found in the plasma, where it goes up 800%. And then if you look at the brain of the animal, you see a massive reduction of plaques and A beta. And of course, I'm not going to show you that. Again, in the paper, it's all reported. You see an effect in cognitive decline, and as effectively we create an anti-Alzheimer drug. So to conclude this first part of my talk, what I'm showing is by simply putting 1+1 ligands into a supramolecular object, accounting for all the different interaction that biology offers, I can actually create a completely new way to create therapies where you display supramolecular materials to transform information about the cells into proper precision therapy. And this is really exciting. And hopefully one day we can produce some really very groundbreaking medicine. Now, to go more into biology things, the other important element in emergency property, and something that we've been studying, is trying to get away from simply Brownian motion. If I put a drug or a nanoparticle, a supramolecular drug, if you allow me that word, into solution, that things will be simply buffeted around by the water Brownian motion. But cells and biology do this with much more purpose. There are many elements in biological world that show actual movement, action towards a specific direction, often driven by chemical gradient in a process called chemotaxis. What I'm showing here is a video of T cells. T cells are our immune cells, they are able to move using various movement processes. In particular, what I'm showing here is apoptosis, something that moves through adhesion, and they recognize its target. In this particular case, this cell gonna reach a cancer cells, and eventually, once it reaches objective, it just will grab it and kill it. And that is, I think, an exciting opportunity if we can mimic that kind of sense of purpose into our system. Now, lucky for us, there were clever people in physics, in particular a colleague of mine, Ramin Golestanian, which actually shared, we worked together when we were both at Sheffield in UK. We started in the same place. It shows something very interesting mathematically, that if I have a particle, let's say a colloidal object, could be a vesicle, could be a nanocolloid, it could be a microparticle, whatever, where you change the surface such as a single patch of it is reacted towards even a given substrate, you start to observe something very interesting. The particle will move more towards or away that chemical gradient. This is a process called self-diffusophoresis. This has given birth to what we call now active colloids. And there are so many elements that are being proposed. So many review there. I'm using one of my colleagues from IBEC, Samuel Sanchez, which shows how you can create that kind of activity in a symmetric version using various different methods of materials design. Now, what I'm going to show you is how we can do this into a more mimetic way. And again, going back to our self-assembling system using either liposomes or polymersomes, vesicles. These are effectively, and I don't need to tell you, these audits in particular, these are always often seen as a very good example to construct what is called a protocell or mimic a minimal cells. Liposome in particular, and more recently, the synthetic alternative made of polymersome, they have this ability to encapsulate certain amount of water-soluble element into a very defined space, and therefore creating this chemical gradient that's necessary, if you like, to drive life, etc. Not surprised, liposome and polymersome have been proposed and designed in order to create a sort of activity to find out whether these things can move towards a certain gradient. Either by chemotaxis or similar behavior. And there are many work from Ayush Mansen from Penn State, Anila Wilson from Rambon University, and my colleague at the IBEC, Samuel Sanchez, show also similar stuff. Now, in my group, like I said, we have this, how we tackle this problem using our approach of creating this sort of positions of assembly and creating patterns in those systems. In particular, we want to create asymmetry. We want to make sure one part of the vesicle is different from the other part. And again, we do that by mixing different block of polymer together. And not, without going into a sophisticated pattern, we want this sort of Janus-like type of structures. And so the idea is to create a vesicle that one membranous compartment is one polymer, and then we have this little patch of another polymer. And the reason we want to do that is because with polymer chemistry, we can choose polymers that have a very different rate of permeability towards a given substrate. And the idea was to encapsulate an enzyme inside these vesicles and make sure that our enzyme substrate react with it, but it will be preferentially passing in and out as a reagent, as a product, through the patch that is more permeable. And so we chose a system, this is really all work now, where we chose a polymer where we know self-assemble with enough hydrophobicity to form our membrane, but very hydrophilic, so everything gets through this PBO polymer, and then the rest will be very hydrophobic PDPA aliphatic chain, and then we encapsulate two enzymes, glucose oxidase and catalases, where we show that this combination is very good, especially for biological application in mind, because we want to use glucose as a fuel. Then glucose oxidase transforms glucose into hydrogen peroxide and glucolactone. But hydrogen peroxide can be a bit toxic, so we add the catalases to remove the hydrogen peroxide and actually feed oxygen into the glucose oxidase, and then creating water as a substrate. And this is great, so it's a very, very safe chemical cascade. And, very old work now, we show this system works. I think one of the best data we had back then, it was this Petri dish experiment where we filled up everything with fluorescence labeled polymersomes and added right in the middle some glucose. And when we have the right combination of enzymes and patchiness, etc., what you see as soon as we add the glucose, the particles start to move all towards the center where the glucose comes from. Why, when you have a system which is fully symmetrical, nothing changes. There is a little bit of disturbance due to the droplet of glucose falling, but nothing more. Now, you can apply this in the context of biological system, a little bit of fluid dynamic diversions. If I allow me to represent a capillary or a blood vessel like a simple pipe, something interesting emerge when you start to add something into this pipe. And also, actually, when you have a small particles, the smaller the particle, the more it will be diffuse, less it will diffuse inside the pipe. Anyway, this is called Taylor-Harrison. And the reason I'm showing that is because we exploit this process to exploit this kind of gradient. And then back in the days, we injected this into the animal, into a rat in this case, and then exploiting the glucose gradient that is always present in the brain to actually, because the brain is very glucose angry, and so exploiting this sort of a Taylor's series dispersion to then allow our particle to spend more time on the barriers and the capillary side. And the result was very nice because the particles that were active were able to get inside the brain more effectively than the particles that weren't active, etc. Now, all of this becomes a very complex physics problem because effectively we have a particle which is very tiny, so it rotates really fast. Yes, there is asymmetry. Yes, there will be a flow across this patch more than the other part. You can model this. What you have, however, is not really a typical rocket type of system. You have a system that continually will rotate, but when it's facing the gradient, the rotation is different, and then there is a slightly movement, so kind of a pulse in there. So effectively, your particle is not really rocketing towards the gradient. It actually will go through this sort of bias random walk into that, etc. Now, this gets complex very quickly because it depends how you have your gradient and you create your gradient to your system, how your reaction occurs, how fast the reaction is converted. There's some really nice physics out there presented by the group of Lowen against my colleague Ramin Golestanian. They show that you actually can create this chemotaxis behavior where the particle really shuttle all the way. You have this sort of polar run and tumble, Ferretti response, etc. We in our side, because we work with very, very small particles, we tend to have this sort of Ferretti responses. Now, this is also experimentally very challenging, and trying to capture the dynamics of small particles is actually very difficult. Most of my colleagues and the data they show you from my colleagues across the world, they tend to work with microparticles. They're very easy to see in a microscope, and they can create a lot of nice tracking. We want to work with particles which are smaller than 100 nanometers. And so the only way we were able to do this is by using fluorescence. So we start to capture this sort of pixelated fluorescence distribution of our particles, each of them labeled with a specific probes, and then measure the distribution of displacement in over thousands and thousands of population. It is important that this number 1,000 stays with you because the statistics is very critical in this element. Because at the end of the day, what you see is a shift of distribution. You don't really see a single particle moving because statistically you need to have a lot of population in order to do that. And then one way we did it is by creating a sort of microfluid device where we create a gradient from A to B and then imaging things. This is the section across right here where the gradient is the more linear, the easiest one to predict. But turns out this is not really never easy. And again, this sort of gradient creates some very interesting behavior. Already, if you have too much of a gradient, you start to have a density variation between one size, and that means that there is flow going actually also in Z. So that the gradient is not that linear. And more important is that to see advection. So it's very important you capture all of this dynamic, etc. And another interesting element is a process called diffusophoresis, where as the molecules comes on one side and react to the surface, it starts to create a completely different functionalization of the surface, and that changes the property of water, how the water interacts with the surface, and again creates some sort of flow. So it's very important you capture all of these dynamics and you take in consideration when you do that. Fortunately for us, we were helped by a lot of very clever physicists and theoreticians that help us to really appreciate this sort of process. The beauty of this process called diffusophoresis, however, is entirely driven by chemical interaction. So how the molecules that represented by this yellow dot here interact with the surface and create a specific bias of the gradient in Z to create a flow that either goes that way or that way, depending where the molecule likes the gradient, so there is an attraction, or whether there is a repulsion. And this is very nice because effectively allows us to really start to use this fluid dynamic exercise in order to measure very accurately chemical interaction. long story short, you add and really characterize your stuff into your system, and every time you change your gradient, for example, I'm going to give you a simple example with glucose. You have a certain flow dictated by the interaction. Well, for example, sorry, if I look at the density gradient, when I work with very high gradient, it doesn't matter that the chemical species, glucose, urea will create the same sort of fluid dynamic patterns. But if I look at the small gradient, in this case 5 millimolars, now the differences between glucose and in this case urea in the same element, it will change. In one case you have a flow like that, in the other case you have a flow like that, which means urea will be repelled by the surfaces while glucose will be attracted by the surface. So you have either active or negative or positive diffusophoresis. So this is just to tell you that simply tracking the particle becomes very complicated. Now making the particles is also very complicated, because if we keep using this sort of patchy approach where we simply mix polymer together to create this phase separation and then create these sort of patterns on the cell, on the particle, really, I show you some nice asymmetric vesicle, but if I show you all of them together, you keep seeing them, but you also see particle with 2 patches, particle with 3, 4 patches, etc. This is a very uncontrollable process. And then we thought, how this is possible in living systems. So we decided to go the other way around and try to break down these elements that we learned from all of these experiments we done in trying to mimic this behavior using now completely stay away from synthetic materials and using natural materials, in particular lipid vesicles. And so we came up with this idea, actually, when you create a lipid vesicles, not only you can encapsulate enzyme, you also have the possibility to house membrane proteins into the membrane. And this is interesting, actually, because many of these membrane proteins have a very, very specific role, which is that one of transporting molecules from one side to the others. So we thought, actually, to create a very, very simple vesicle where we have our lipid vesicle, we encapsulate an enzyme, glucose oxidases, ureases. There are many different enzymes we can choose. And then we chose this protein called alpha hemolysin. This is a toxic protein secreted by certain bacteria, which has the ability of forming these really nice nanopores across any type of lipid vesicles. It's very unspecific. It simply creates a hole effectively across the membrane. And now what we want to do with it is to create a system that is now that has an enzyme inside that reacts to this given substrate, and now this hole allows the transport of the substrate and product across. And the beauty of this is that simply by playing with the stoichiometry of our pores, so in other words, trying to make vesicles that have 1 pore or no more than 2 pores per vesicle, then inevitably you start to create permeability. This is what I'm showing you is the calcium assays where we measure how the calcium react with the impermeable probes as we increase the number of pores. And then what we did, we added just enough in order to create a symmetry. And then again, we start to look our horrible microfluidic device to capture this dynamic, measure thousands and thousands of particles, discover that you actually start to observe some very, very complex behavior, and then you need to track your particle in different Z, not just in one Z. And again, you start to observe a very beautiful active process happening either chemotactically towards the gradient or away from the gradient. And most importantly, we see this with both glucose and urea by using either glucose oxidase and ureases. And this is really nice because effectively allows to show that this process is kind of universal. As long as I have a vesicle which encapsulates a given enzyme and then it has a membrane pore with a number which is very close to 1 effectively I create a symmetry and therefore I create a kind of a control activity. Now, this is our contribution to a field that is now exploding in many different ways. Now, most of my colleagues don't even call it active matter anymore. They are now claiming the word nanorobotics. And there are many people in the world, and they are trying out to show how this can be used for many applications and really creating that kind of a fantastic voyage, etc. And I think I really want to hear you too. So I decided to not give too many details and allow for more Q&A sessions. And so I'm going to stop here with my presentations. Again, perhaps going back to my fantastic team that has allowed me to show you all of this data, and particularly The data I show you is Lorena helped a lot in understanding of the materials from a TM point of view. Valentino and Barbara, who's now finishing her PhD, will be next week. Defense was the first author, and then many people that were survived my research group and my short attention span and a big chaotic supervision styles. And now I've found a really good job and very pleased with how these people move on into their career. And of course, the founders and you all for the attention. Thank you very much.
Thank you. That was amazing. I just want to say not just the topic, but also the presentation. Your slides are breathtaking. There's quite a bit of questions in chat. Can you see chat?
Sure, I can open it. Yes, I got it.
Can you read the question out loud because people watching the recording don't see the chat?
Right. So I'm just gonna. The first one is something to do with reconstituting destroy US science. I think that's a question not for me. Uh, I'm afraid. Yes. Okay. There is a question from, as far as I see, the key lock cell model, you use triple compartment model. What about choosing two times biomarker compartment in and a biological circuit structure? Would it be sufficient for cancer? Thank you for your answer. Yes, of course, the more complexity we can add, the more specific we become. But also, to the point, you need to be careful. You start to lose universality, because as you start to get that kind of level of selectivity now, and this is where we are now talking with computational science to help us out, you start to almost become personalized. So the drug is no longer working across the population. You start to make it patient specific. And this is what we want eventually, ultimately, to create a kind of level of personalization that really precision medicine can deliver. But yes, the more complexity, the more this biological circuit structure and then understanding transport with ligands, etc., you become more and more specific and more selective and therefore more potent. I don't know if I answer your question, Oleg. I'm going to move on to the question from Anna. I appreciate the physics grounding of your design. Could we use this principle to design residence time or receptor binding completely in vitro for synthetic cell line design? And the member, yes, absolutely. As soon as you have multivalencies, residence time, it goes really long. Because statistically, the more bonds you form, the more time you will take to break them. And of course, as the one breaks, the other one forms, etc. So the K-off collectively becomes smaller and smaller and smaller. And so then you really can create some very indefinitely long time. Then I have a question from Harry Taylor. Can we add the non-canonic building blocks to your pipeline? Could you use, for example, 20 more amino acids in a protein design? Yes. I mean, I would love to actually work with protein design. This is an area that we want to explore more at the moment. For me, we know we need to make block of polymers, and we work with them. And so it was easier. And but remember, our ligands is a peptidic materials, typically 11, 20 amino acids, sometimes 5 or 6, depending what receptor we have. But we combine this peptide with our synthetic material. And then I have a question from Trisha. Can the key-lock model be used to model something more complex? For example, how to cell interact and all the Yes, it is for me, and actually, hopefully, very soon I will send this paper out. It's a very universal process. Multivalence is not just simply for drugs. I think for me, it's easier to sell the drug because that's what we want to ultimately make. But effectively, we actually, for example, published a paper where we showed the same process applied to viruses, how virus enter cells. And of course, it is how immune system works. In most cases, and already at the molecular level, IgG and antibody in general have more than one bonds. Possible IgG, the simplest one, has 2 epitope binding. IgM has 10 epitope binding, etc. And in the middle, you have other different morphology. And then on top of it, as the more antibody binds to its target, you create multivalencies and then how macrophage recognize them and then create certain responses. So this is, multivalence is universal for every type of cells. And actually, for me, it's important this idea that as we increase the number of bonds, we switch off the interaction because of the sterics, and actually it's kind of a negative feedback. So because instead of activating a bond because you form the bonds, you actually can switch off certain type of repulsion as well. So you want to do more on that on the other side. I think I got a question from Davis. Can you design more promiscuous enzyme with this model, enzyme with multiple substrate? Yes. You need to have that trick. In order to get that kind of selectivity towards number of receptors, or number of your cells, whatever you want to target, you need to have the single bond needs to be weak. If it's too strong, then you need to go on the other side. You need to have enough and really large amount of bonds in order to create selectivity with sterics. But then you need to have some sort of negative bond, if you like, repulsion to stop that interaction. And I think I read all of them. Yes, I don't think I haven't missed anyone.
Yeah, I think that's oh, Taesok, you have a question?
Okay, so great talk. I want to ask some, I don't know, philosophical question. That's why I don't want to type into the chat. So you
Sure.
showed at the beginning, by the way, great talk. I love it. At the beginning, you kind of talking about entropy increase and then, non-linearity, etc. in biology. And the
Yeah.
question I want to ask is, very from the physics point of view, what do you think, governing principles in biology, and if we have, and I have the
Yeah.
multiple discussion, probably more than 50 people in the world, but I still
Hmm.
figure out clearly. And if you have one or multiple, I just want to hear from you, and how we
Yeah.
gonna use that principle to, for potential application in the future.
So entropy is in many ways the major driving force in our body for a lot of things, right? You do have a heat exchange. There is enthalpies play a very important roles, but one of the most beautiful things is that even though we have such a crowded environment inside our cell, even our tissues are so complex, etc. You create some very beautiful level of the and then again, as you create interaction, you start to create emergency because you start to have this sort of 1 + 1 plus something else that come from the combinations, etc. This is really emergent, etc.
No, no,
And then
I agree with you, but
yeah,
entropy is,
yeah,
to me, is more physical principle or
yeah,
principle physics or
yeah.
chemistry than in biology. I want to ask what would be unique principle if we have, or if any, in unique in biology. That's actually my question.
I think biology is physics for me. There is not really, I mean, however, we do not have yet the mathematics and the physics, theoretical physics model to fully capture the complexity of biology because still physicists are still arguing how to understand three-body interaction, right? And
Mm-hmm.
in biology is much more than three-body, but there is so much you can learn from physical principle in order to understand biology process. And there is a branch of physics called non-equilibrium statistical physics or complex dynamics, which allows you really to capture some of these biological elements and really step away from the traditional linear process.
Mm-hmm.
It is normal, and this is, in my opinion, personal opinion, that in biology, because we deal with complex systems, we need to specialize in understanding how the single element of this complex system works, and we really spend a lot of time understanding the parts. But then how these parts come together often is forgotten. And then this is where something new emerge. And this is what I really want to emphasize in this. And I show you, for example, the example of multivalencies as a great example of that. I just multiply the bonds by two, I create a completely different way of interaction. And then it's definitely lose linearity completely and very quickly. But then linearity is comfortable because allow us to easy prediction. But actually nowadays with machine learning, we can deal with nonlinearity much more. And we can even deal with multiple dimensions, etc. So I think there is I try to call for arm for biologists to start to really look at what physicists are doing and really implement this sort of new physics. Well,
I love
new
that
physics,
answer.
more
So thank you.
the new physics, more advanced physics to understanding biology.
Yeah, so
Yeah.
that confirm my feeling. That means
Mm.
I have been doing art for 30 years rather than doing engineering, even though I claim myself bioengineer. So I'm artist, I guess.
Uh,
Kate is artist, I guess. So thank you. That's a wonderful answer. Thank you.
I actually want to be an artist. That's probably why I spend so much time in my slide design, as you can understand.
Thank you.
Yeah,
All right.
fantastic. There is
Your
another
slides
question from
are
Lindsay.
gorgeous. Yeah, I see there's another question. Yeah.
Thank you. Thank you, Kate. Great. So how do you train your models? Is purely chemical info? Do you need specific condition experimental data? Yes, you need to have the chemical information from your protein and ligand, which is traditional drug discovery exercise, but you also need to know where your receptor sits, in what cells, how many copy of this receptor you have possibly, and more importantly, whether you have similar receptors. And an element that which I really touch very quickly, and we spend a lot of time, is called the glycocalyx. These are the sugars that coat the cells. And these sugars have a very high level of repulsion towards most molecules. So for us, it's an important element when we design. So when we target our cell, we take the three information we take: the receptors, the concentration of receptors, and then the glycocalyx. And then we go and start to build our model around those.
Awesome. Yeah, I just want to say I really appreciate that you're able to kind of shorten the typing experimental pipeline, that the modeling is actually predictive, that it helps us.
Yeah.
To me, that's the key. Okay, it looks like there are no more questions, so I want to say thank you so much again, Beppe, and thanks everyone else for the discussion. And have a great.
Thank you, guys. It's been a great discussion. Nice questions. I hope if you come by Barcelona, please pass by and ask me the question in person. It would be fantastic to see each other in 3D. And of course, feel free to contact me by mails anytime.
Yeah, Barcelona is wonderful. I was there a few weeks ago. Amazing place.
Yes, I cannot blame. I cannot complain, especially after I lived 22 years in the UK. Believe me, I think I deserve Barcelona.
So much. All right,
Yes.
thanks, guys, and have a great rest of your Monday. Bye.
Thank you. Bye. Take care. Have a great week. Bye.